Hello, everyone, and welcome to Lecture 9 in our General Psychology course. And it's the second lecture in our unit on the senses. So last time we talked about senses in general, and we talked about our visual sense. This time we'll be talking about some of the other senses. So hearing, touch, smell, taste. So we'll be learning about those specific senses. Our sense of hearing, which is used to detect what are effectively ripples of air pressure. We call them sound waves, but it's really vibrations in the air, which of course is very different from our sense of touch, which is physical pressure on our skin. And as we'll see, touch is more than just that. We tend to think of our sense of touch as simple pressure on the skin, but there's more to it than that. We also have our senses of smell and taste. And those are very related because they both sense chemicals in the environment. So in particular, what we're going to do is look at sound, first understand what sound is, and then understand how it's processed. And then how that processing, some of which happens in the ear, some of which happens in the brain, how that processing then gives us perception. What are the aspects of perception of sound that have to be considered? We'll also see that, as I mentioned, touch, which we refer to with that one name, actually has a lot of different components. We build all these different sense organs into our sense of touch, and some we think about and some we don't. Things like temperature is arguably part of our sense of touch. But it's not usually what we mean when we say touch. Pain is it part of our sense of touch? I don't know. It's embedded in organs in the skin, so arguably it is part of your sense of touch. But again, it's not what we think of when we say our sense of touch. And then we'll understand how the body and the brain detect these different chemicals. And we call these things smells and tastes. But all they really are different concentrations of particular kinds of chemicals. And so we'll talk about that at the end of today's lecture. So first we'll start with our sense of hearing, otherwise known as audition. And before we get to how hearing works, we first need to understand what sound is. In our previous lecture, we looked at what light is, and so we'll do an equivalent maneuver here. So what is sound? Well, sound is just waves of air pressure. So air molecules are everywhere, there are lots of them, and when a sound occurs, what's really happening is that the source of the sound is pushing air molecules around. And if that source is vibrating, for example, it's pushing them back and forth. And so you get these waves of air pressure that propagate around the environment. And as we do with light, we'll talk about a couple of the properties that sound has. So first, there's frequency, which is measured in peaks per second, otherwise known as hertz. Uh, and so frequency of sound is what we perceive as pitch. So high frequency sounds we call high pitched, low frequency sounds we call low pitched. And what that really describes is how many peaks of those air pressure waves we get per second. And it turns out wavelength and frequency are mathematically related. Just for historical reasons, we describe light in terms of wavelength and sound in terms of frequency. But we could easily do either. Then we have amplitude, which is the height of the peaks, just like it was for light. Now, here we refer not to brightness when it comes to amplitude. Here it's loudness, which is typically measured in decibels. You may have heard of that term before, decibels. And so decibels are a unit of measurement for how loud a sound is, the amplitude of the sound. And of course, if a sound emits a lot of decibels are associated with a particular sound, that means it's really loud. And if it's loud enough, it can damage your hearing. And of course, similar to the purity of light, we also have the complexity of sound. So a pure tone, like you would hear with a tuning fork, will have a single frequency. But any more complex sound will have a number of frequencies. And this is what musically, at least, is referred to as timbre. So the mixture of frequencies that are present in a sound describe its complexity. And so certain musical instruments sound different. They can play the same note, but we can tell which instruments are playing what. If a violin plays a C sharp and a saxophone plays a C sharp, those are the same fundamental note. What changes between the two instruments is the mixture of other frequencies that you're getting. And so that's how you can differentiate between the two. So there's what the sound is, and we can describe it in terms of its frequency, its amplitude, its complexity. Of course, another important part of the sound is where it comes from. What is the source of the sound? And evolutionarily, that was very important. If there was a sound that told you a predator was nearby, the direction of the predator, or prey, if that's what you're doing, those are both important pieces of information. The identity of the sound, but also where the sound is coming from is critical. You don't want to run toward a predator, and you don't want to run away from prey if that's what you're chasing. So the location becomes very important as well. And it's not as easy as it is for vision, where the source of the stimulus has a very clear physical analog in space. A sound can come from anywhere, and it's not obvious where it comes from. It takes some processing. And there are two main clues that we get when it comes to knowing where a sound comes from. One of them is a difference in loudness, amplitude. Uh, and so if a sound is coming from the right, it will be louder for our right ear than for our left, mainly because our head will block out some of the sound. And so there's not as much left to reach our left ear if the sound has already passed through or around our head. It gets directly to the right ear. And so you have this difference in loudness. Obviously, conversely, if the sound's on the left, it'll be louder to the left ear than the right. The other way to tell where sound is coming from is subtle differences in timing. And so if there's an object on the right emitting a sound, that sound will reach the right ear first. And that may seem like a trivial difference. But in fact, sound, as fast as it is slow enough that your nervous system can pick up on differences in timing. Light is way too fast. Your system cannot tell whether light hits your right eye first or your left eye first, because light's just way too fast. But sound is much slower. And so your nervous system can actually tell differences in timing. So if a sound is on the right, your right ear will get it first, and your nervous system can tell where that difference in timing lies, how big the difference is, which tells your system how far to the right it is. Not in terms of overall distance, but the angle. If it's straight ahead, it'll reach the ears at the same time. If it's slightly to the left, it'll get to the left ear a little bit sooner. If it's way off to the left, it'll reach the left ear much sooner than the right. So that's what a sound is. That's how we localize sound. Now, when it comes to the auditory system, first we need to talk about the anatomy of the ear. So here in the lower left, we have a diagram of the ear, and the ear has three sort of sections. First, there's the outer ear, which I've highlighted here in blue. And the outer ear, the outermost part, the part we see, the part we might refer to as someone's ear, is what's called technically the pinna. So that pinna is that folded structure of cartilage and skin that is the outermost part of the ear. And the pinna is shaped that way for a good reason. It helps to filter sounds. Technically, it's another way we localize sound is because of the filtering properties of the pinna. We don't need to go into details there, but it does have a function. The eardrum is the innermost part of the outer ear. So you have the pinna, then the sound passes through the auditory canal, and then you have the eardrum. And the eardrum is a membrane that separates the rest of the ear from the outer ear. And most importantly, when sound waves come into the outer ear, they make that eardrum vibrate. And of course, sound waves, as we just discussed, are waves of air pressure. So you have air molecules that are hitting the eardrum sort of in an off and on pattern. They'll push, and then they'll not push so hard, and they'll push again, and then not push so hard. And that frequency of those air pressure waves moves the eardrum back and forth, makes it vibrate. And then what that does is activate the middle ear. And the middle ear is pretty small in terms of volume. Uh, its components, its main components, are what are called the ossicles. And you may have heard of the hammer, the anvil, the stirrup. They have Latin names as well. We're not going to belabor the names of the little bones, but the ossicles are three small bones in the middle ear. And what they do is they convert those vibrations of the eardrum into vibrations in the fluid in an organ of the inner ear. I'll talk about it in just a second. But these ossicles are set up to amplify the vibrations in the eardrum. And so those vibrations then get passed on, and it's the inner ear, the main part of which that concerns us right now is the cochlea. Uh, there's fluid inside the cochlea, and those ossicles make that fluid wash back and forth. Of course, vibrations, especially sound waves, tend to be pretty fast. So it's not visibly washing fluid back and forth, but it's moving it at the same frequency as the sound waves. And so we'll talk more about what goes on in the cochlea here in just one second. But the cochlea is the main part of the inner ear, and you can see it highlighted in blue. It is the lower right portion that's highlighted in blue. It looks kind of like a snail shell. Once sound gets processed by the cochlea, it gets passed along the auditory nerve and then goes to the brain. And first it goes to primary auditory cortex, sometimes called A1. So last time we had primary visual cortex, called B1. This is primary auditory cortex, A1. And rather than being located in the occipital lobe, A1 is located in the temporal lobe. And it has an interesting feature. So it's mapped out, auditory cortex, according to frequency. Different parts of auditory cortex will become active depending on the pitch of the sound being heard. And so high-pitched sounds cause activity closer to the parietal lobe. Low-pitched sounds cause more activity out toward the end of the temporal lobe. So there's sort of this frequency map. The brain encodes the frequency, the pitch of the sound, according to where along the temporal lobe activity is occurring. And there seem to be two pathways in auditory cortex, similar to what's going on in visual cortex. This has not been known for as long, it's not as ironclad, but the caudal end of auditory cortex, caudal meaning toward the tail, human beings don't have a tail, of course, but toward the back of the head for us, there's the Ware pathway, similar to the dorsal Ware pathway for vision. Here we have a caudal Ware pathway toward the back of the temporal lobe. And these are areas that process where a sound is coming from. And in contrast, you have the ventral what pathway, similar to the ventral what pathway for vision. And so down toward the lower end of the temporal lobe, you have portions of auditory cortex that are responsible for identifying a sound. What is the identity of the sound? What's producing it? And so again, we don't have as much data on this phenomenon as we do for vision, but there does still seem to be this pattern. Okay, so that's sort of the overview of the anatomy of the auditory system, but we still haven't covered how we get from sound waves to neural signals in the brain. And for that, we have to understand the cochlea. And the cochlea is that snail shell-shaped organ found in the inner ear. And if we look at it the inside of the cochlea, we'll see what's called a basilar membrane, which is a piece of tissue, and it's swimming in fluid inside the cochlea. Now, if you unroll the cochlea, unroll that snail shell, you'll see that the basilar membrane changes in terms of width from one end of the cochlea to the other. What you'll also notice is that the basilar membrane, embedded within it, are what are called hair cells. These are specialized neurons that fire whenever the basilar membrane moves. Now, it turns out the basilar membrane has a bunch of hair cells all along its length. And it turns out that depending on where the basal membrane moves, only those hair cells will become active. And if they're active, then they send action potentials along the auditory nerve to the brain. So how does this work? How do we get from sound waves to those neural signals? So whenever sound waves hit the eardrum, it vibrates. And when the eardrum vibrates, then the ossicles, those bones in the middle ear, vibrate as well. And they move back and forth, and what they do is push on fluid inside the cochlea. And so that fluid starts moving back and forth. Now, when that happens, the basilar membrane moves back and forth along with the fluid. Sort of like seaweed in the ocean. As waves come and go, the seaweed will move back and forth. Same idea here. So the basilar membrane moves back and forth, corresponding to those vibrations that the eardrum is getting. And then what's interesting is that the basilar membrane is not the same along its entire length. It actually gets wider as you move out toward the end of the cochlea. And what that means is that the lower the pitch of the sound, the more active that far end of the basilar membrane will be. It'll move more. If it's a high-pitched sound, the end of the basilar membrane near the base of the cochlea will move more. And so depending on what the pitch of the sound is, you'll get movement in different parts of that basilar membrane. And that is what's called the place code. So a different part of the basilar membrane moves. It moves those hair cells along with it, and only those hair cells will cause action potentials. And so there are neurons in the auditory nerve that correspond to different locations on the basilar membrane. So that's how we get a stream of information to auditory cortex that says something about the pitch of the sound. Because what it's really doing is indicating which location along the basilar membrane is moving back and forth. And so we have locations along the basilar membrane and locations along the auditory cortex that become active together and indicate what pitch is being played. You also have a temporal code. And this is something that occurs for low-pitched sounds. Neurons can fire up to around 1,000 times a second, maybe a little bit more. And the frequencies of sound that we can hear as humans can get down to 100 hertz, pretty low. They can get up to 20,000 hertz, though, so it's a big range. But for those lower pitched sounds, neurons can actually fire fast enough to indicate the frequency. Now, once you get up past 5,000 hertz, the neurons can't keep up. But for those lower pitched sounds, those lower frequencies, neurons can fire and match the frequency of the sound that's being played. Okay, so that's how sound gets transduced to neural signals. Sometimes, of course, individuals suffer hearing loss. And there are two main kinds that we'll talk about here. They are conductive and sensory neural. So in conductive hearing loss, sound waves are not getting to the eardrum, or they're getting to the eardrum, but the ossicles, for example, have fused together, and so they don't move along with the eardrum. Either way, you're not getting vibrations in the fluid in the cochlea. So, of course, the basilar membrane doesn't pick up those vibrations. And that's conductive hearing loss that can often be remedied by medicine or by surgery. The other kind is sensory neural, and it's a little more complicated. And what's happening there is there's either something going on to the basilar membrane, or hair cells have died, or neurons in the auditory nerve have died. Any of those things can happen and cause signals not to reach the auditory cortex. And the way that is addressed is often with a cochlear implant, which we see in this picture here. And this is a device that picks up sound, and it has one end of the device that's been surgically implanted into the cochlea. And that end of the device has electrodes on it. It will stimulate hair cells or neurons in the auditory nerve. It'll stimulate those cells so that they fire. And so instead of the eardrum, ossicles, basilar membrane causing that stimulation, you now have this device that, depending on the frequency of sounds, will then cause the stimulation of the auditory nerve. And so cochlear implants are relatively new technology, but are often very helpful for those that have hearing loss. Okay, so that is it for the auditory system. Now we'll move on to our sense of our body. Uh, as we'll see, we call it often our sense of touch, but that's not entirely accurate. And so what your body senses do is tell you what's going on both inside and outside of your body. It gives you sort of the state of affairs of your very local environment. So it's not just your sense of touch. It's not just pressure on your skin or vibration on your skin. There are other things going on as well. So it involves multiple sense organs. So for the sense of touch itself, you have sense organs for different aspects of that sense of touch. Things like pressure. So something pressing on your skin. There are a set of sense organs dedicated to that. Vibration is handled by a different set of sense organs. So feeling pressure against your skin, if something's poking you, is different than feeling very high vibrations, like texture. So when you run your hand over velvet, or you run your hand over felt, or you run your hand over a smooth surface, you're putting the same amount of pressure on those surfaces, but tiny vibrations due to the texture of those surfaces can be detected. So you can tell the difference between those textured surfaces. Of course, we also have temperature. Temperature is handled by a different set of sense organs embedded in your skin. So there are all these different sense organs that contribute to your sense of touch, so to speak. What's interesting is that when we get to the brain, we have what's called contralateral organization. And what do I mean by that? Well, it turns out that the right side of your body is processed in terms of touch by the left side of your brain, and vice versa. The left side of your body is processed by the right side of your brain. Uh, so we have this contralateral, it means opposite side, organization. And that's a theme that pops up again and again in the nervous system. There are various theories as to why it's organized that way. We don't know for sure, but that's the way it is. In addition, the part of the brain, which is in the parietal lobe, that deals with sense of touch, exhibits what's called somatotopy. What does that mean? Well, it means that the cortex in that area is laid out according to the map of the body. So soma means body, and topi means surface. And so what we have is this gyrus of the brain, where part of the gyrus corresponds to the hand, part of the gyrus corresponds to the leg, part of the gyrus corresponds to the lips. And so when those parts of the body are touched, when they have sensation, you'll see activity in the corresponding part of that gyrus. Also, if you're manipulating the brain, if you're performing brain surgery, for example, or just stimulating it electrically, activity in that part of the gyrus, if you induce it, will cause the person to feel as though pressure or vibration is happening on that part of their body. So if you electrically stimulate the part of the brain that corresponds to the hand, it feels as though someone is touching your hand. So the touch is organized according to the plan of the body. Okay, so that's our sense of touch as we usually think about it. Certainly pressure, vibration, we build into our sense of touch. Temperature, maybe you do, maybe you don't. But temperature is certainly handled by sense organs in your skin. Another sense that's part of your sense of touch, really, is pain, at least pain in the somatic part of your body. Your sense of touch is embedded in your skin, as are receptors for pain. And of course, the question is, what is the purpose of pain? Why do we have it? Well, pain, as unpleasant as it is helpful in many circumstances. It's a warning that your body is about to suffer some sort of damage, or maybe that it has suffered damage, and that you shouldn't stress that part of the body more than you already have. So pain does have a purpose, and you might think it'd be great to live without pain. There are people who are born or suffer the loss of their sense of pain, and it's very difficult. They end up incurring injuries that they're not aware of and making them worse, especially small children. So the sense of pain um, is actually quite useful, which is not to say that it doesn't malfunction sometimes. People who suffer from chronic pain obviously have more pain than they should, but having a sense of pain at all is a useful evolutionary adaptation. And you have specialized neurons for different kinds of pain. There are what are called A delta fibers, um, and there are C fibers. And these are different kinds of neurons in your spinal cord, for example. And the A delta fibers handle that sharp sense, the onset of pain, and the C fibers are that dull, that ache that happens after a painful stimulus. So those stimuli feel different, and in fact, they're handled by different neurons with different properties. And drugs have been developed that act on one kind of fiber and not the other. And in fact, pain, of course, is a big area of drug development. And these different kinds of neurons, different kinds of pain receptors, different locations of neurons that deal with pain are all possible targets for specific drugs. What's interesting is that pain has two separate pathways in addition to these different kinds of neurons. There's what's called the sensory pathway, which is just your body's way of registering where the pain has occurred. And that goes up to somatosensory cortex, that gyrus in the parietal lobe that registers other kinds of touch. But there's also an emotional pathway. And this set of neurons goes to things like the amygdala, which deal with emotions. So you have an emotional experience of pain. It's not just simply registering where your body's been hurt. There's an emotional experience. It also goes to your hypothalamus, this emotional pathway. And the hypothalamus deals with hormonal control. And so it's sort of a two-way street. Your body can actually affect the experience of pain, but so can your brain. And the hypothalamus is a key player there. And this gets to what's called the gate control theory of pain, the idea that neurons in your spinal cord will send pain information up to the brain, but that those impulses can be blocked or diminished either by stimulating that same part of the body. So if you stimulate the sense of touch in that same part of the body where the pain is occurring, the pain may be diminished. So that's sort of what we call the bottom-up pathway, going from the body up to the brain. But your brain can also inhibit pain signals, so from the top-down approach. They can tell your spinal cord, don't send as much information about pain. And this is true when, for example, you're in a stressful situation, your sympathetic nervous system has been engaged, and that's where the hypothalamus comes in. So if you're in fight or flight mode, if you're trying to run away from something or you're engaged in physical combat of some sort, you don't want to worry about pain. You don't want to deal with the distraction of a painful stimulus. And so your brain can actually get your spinal cord to send less information about pain, to weaken that signal. You have other body senses as well. Where your body is in space is a big one. So you can close your eyes and you can still tell how your body is positioned. And this has to do with sense organs in your muscles and tendons. And so you have these different sense organs that can tell how much tension is in your muscles, how much tension is in your tendons. And that tells these sense organs and eventually your brain which muscles are flexed, which ones are not, and therefore what position your body is in. Now, when it comes to things like movement and balance, there you depend in large part on your vestibular system, which we see here in the top right. So your vestibular system is informed by what are called semicircular canals. And these are these loops of tissue you see on the opposite end of the inner ear from the cochlea. So the cochlea is sort of snail shell shaped. The semicircular canals are these loops that are sitting on top of it. And they're filled with fluid, and when your head moves, it moves that fluid. And we're not going to delve into the details, but that fluid movement, just like it is in the cochlea, is transduced into neural signals. So when your head moves in a particular direction, particular neurons will get stimulated by that movement. And that helps your body, your brain, say which way you're moving. So if your head spins or if your head moves from side to side, you can tell that you're moving. And the visual system has a role here as well. If you have your eyes open, you can see your environment. You can see if you're moving relative to your environment. And this is where the concept of motion sickness comes up. So if your vestibular system is telling you you're moving one way, and your visual system is telling you you're moving another, you will often not be able to reconcile those pieces of information. You'll sometimes get nauseous or dizzy. And that's because you're getting conflicting pieces of information. So if you're in a car, for example, your vestibular system, you've adapted to being in the car. It can't tell that you're moving because your body isn't moving relative to the car. It's the car that's moving. However, if you look around, your visual system sees things moving all around you. And these two different sources of information can come into conflict when the car is rocking back and forth, for example, or if you're looking down and not watching the road move in relation to your body. So depending on what your sensory systems are receiving in terms of information, these things can come into conflict. Usually, they all agree and we can tell where our body is and whether we're moving or not, but not always. Okay, finally we're going to wrap up with a discussion of the chemical senses, taste and smell. And so the reason we've lumped them together is that they both detect chemicals in the environment. Now, granted, it's at different distances. Your sense of smell can be used to detect odors from far away, and your sense of taste is obviously in the confines of your mouth. And they're also different in terms of sensitivity. Your sense of smell, even though it's not very good in humans compared to other animals, is far more sophisticated than your sense of taste. You only have a few basic tastes that you can detect. If this doesn't seem right, just try holding your nose and eating a bit of apple. It won't have much flavor to it. And in fact, if you block odors completely, all you really have to go on is texture and these, again, sort of coarse-grained senses of taste. People often can't tell the difference between a piece of apple and a piece of potato if they're holding their nose or if they've got a severe cold. So the sense of smell is more nuanced than the sense of taste, but they both detect chemicals. So first, your sense of smell, which is also called olfaction. So it's your olfactory system is your sense of smell. First, you have odorants, which I've highlighted here at the bottom right. And those are just chemicals. They're molecules that are floating around in the air, and as you breathe in, you draw them into your nasal cavity. And so these different chemicals are floating around in the air inside your nasal cavity, and depending on where they are, and if there's enough of them, they can latch on to olfactory receptor neurons. So these are neurons that actually poke through the part of your skull at the top of the nasal cavity and stick out into the nasal cavity itself. And they've got dendrites that poke out, and they're waiting for odorants to come along. So these molecules will come along and bind to the dendrites, just like in normal neurons, neurotransmitters will come along and bind to dendrites along receptors. So your olfactory receptor neurons, there's a lot of different kinds. They have different receptors for different chemicals. And so if an olfactory receptor neuron becomes active, if it binds to an odorant, then you will experience, if it's common enough, it's concentrated enough, you'll experience the smell corresponding to that odorant. So that's what your olfactory receptor neurons do, is they are the neurons, the sense organs. They actually detect the presence of chemicals. They then convey that information to what's called the olfactory bulb, which is sort of a lump of neurons, a group of neurons, that sits on top of your nasal cavity. It's not inside your brain. It sits on top of your nasal cavity and collects and organizes olfactory information. From there, that information is transmitted to olfactory cortex. What's interesting is that olfactory cortex is not very big. But your sense of smell tends to trigger things like emotions to a larger degree than your other senses do. And the reason is that, first, your olfactory system bypasses the thalamus. It's the only sensory system that doesn't go through the thalamus. Instead, it goes to parts of your brain that are adjacent to regions like the amygdala, hippocampus. So senses, smells can be very connected, very evocative when it comes to emotions and memories, more so than other senses. So smells can be very powerful in terms of calling up a memory or causing an emotional response. And there are particular odorants called pheromones, which you may have heard of. And pheromones are chemicals that are released by members of your own species that affect your behavior. That's what a pheromone is. And so these are chemical signals between organisms of the same species. And pheromone, the question of how pheromones, how important they are in humans is an open debate. There's some limited evidence that maybe they affect our behavior. Many animals have a specialized organ for detecting pheromones. Ours is actually dormant. But there's evidence we may be able to detect pheromones through our typical or usual sense of smell. Whereas other animals, their behavior is highly dependent on pheromones. Ours, not so much, but there may be some effect there. And then finally, when it comes to our sense of taste, most of the sense organs there, of course, are in the tongue. And I say most. You do have taste receptors uh, in the back of your throat and on your epiglottis, which is a flap of tissue that uh, seals off the lungs when you're swallowing. But mostly these things are in the tongue. And there are sort of a hierarchy of structures involved in taste. The first are the papillae, which you see here, which I've circled in this diagram. Uh, and these are the bumps that you see on your tongue. People will often refer to those as taste buds. They're not. Those are papillae. The papillae, however, are covered in taste buds. So taste buds are much smaller than you can see, but you still have a lot of them in your tongue. They're just not visible. And within each taste bud are taste receptor cells, sort of like the olfactory receptor neurons. These are cells that are waiting for a particular chemical to come along and bind to receptors and cause these taste receptor cells to fire an action potential. So that's how we transduce the presence of chemicals into neural signals. Chemical comes along, binds to receptors on the dendrites of these cells, and then causes those cells to fire an action potential. So the taste receptor cells are where the transduction happens. And eventually that information gets transmitted to what's called gustatory cortex. And this is the part of the brain that deals with taste. It's not very big. It's located in your frontal lobe, but it does process taste sensation. As I mentioned before, your sense of taste is not particularly sensitive. It's not particularly varied in terms of what it can detect, but your brain does process it, obviously, and we can perceive tastes. And there are a few different kinds of tastes. Um, at first, we just thought there were four. Then the fifth one was discovered, and more recently, there may even be a sixth. And these correspond to different kinds of taste receptor cells. So there is salty, and this corresponds to mainly sodium. So the presence of sodium we perceive as being salty. So our sense organs, taste receptor cells, sense sodium, we perceive saltiness. There's sour, which is a response to the presence of acids. If a food is acidic, it tastes sour to us. There's sweetness. So if sugars are present in the mouth, taste receptor cells that cater to sugar molecules will become active, and we will detect and perceive sweetness. There's also bitterness. Sour corresponds to acid. Bitterness often corresponds to basic, that is high pH tastes or chemicals. And so if a basic chemical enters the mouth, it can be detected by taste receptor cells, and this is what we interpret as being bitter. The fifth one is called umami, and that roughly corresponds to a savory or a meaty taste. And as far as we know, these taste receptor cells are responding to the presence of things like glutamate, which is one of our neurotransmitters, but it's also present in particularly savory foods, and so that's what we seem to be detecting. The sixth taste molecule, uh, we may have the capacity to taste fats or fattiness, or more accurately, oiliness. This is relatively new, so the jury's still out on that, but that is a potential sixth type of taste. Okay, that will do it for this time. Next time, we will start talking about learning. We'll learn about learning. So we'll start off with simple kinds of learning, like classical conditioning, in which we associate one event with another. Pavlov's dog springs to mind. There's also operant conditioning, where the events in our environment depend on our own behavior. So we'll talk about that learning process. How is that different from classical conditioning, where our behavior doesn't matter? And then we'll talk about the neural elements of learning. How do these different kinds of learning, how are they handled and processed by the brain? What parts of the brain are important? So that will begin our unit on learning, and so I will see you then.